Hello, students. Welcome to Chapter 9 and a little bit of Chapter 10. In biopsychology, we're going to learn about touch sensation, pain, and then a little bit about motor control. So, one quick announcement. Don't forget to check for Psychology Club. Depending on the quarter that you're watching this, it might not be the same time that is on this flyer. So just look around for a flyer for the Psych Club. All right, so let's jump into it. This is your skin. So we have these type of receptors all over our skin. There are lots more in your hands than there are on, say, your torso or your thighs. So, but all of these different receptors get at a different, give you a different bit of sensation. And so putting them all together is how we get our full understanding as we're, say, touching something. So I might be really small on the screen right now, but I'm lifting up a green pen. Oh, hey, it's disappearing the pen on the green screen. That's funny. So as I touch this part of the pen, it's rubbery and there's some like grippy lines, like ribs on it, and then the plastic here is smooth. So how can I tell those things? Well, it's because of these touch receptors. So up here we can see we've got some Merkel's discs over here. We've also got some Meissner's corpuscles right over here. So you can see those are pretty close to the surface of the skin. So basically just under that first layer of skin, we've got those Merkel discs and Meissner's corpuscles. Further down, we have these Ruffini's endings, and we also have the Pacinian corpuscles. So those are deeper into the skin, like in some of the middle and bottom layers of the skin. We also have these free nerve endings. So there's basically axon, or you could think of them as dendrite branches hanging out in the skin, and they are where we get a lot of our pain reception from. So there are ones that are specifically tailored to temperature, some that are tailored to chemical, and some that are tailored for mechanical, so like pressure or if you got a cut. So, but let's go through each one of these in a little bit more detail. Before we do that, let's watch a general video of all of the receptors to get you a good starting point before diving into them in each a little bit more detail.
Touch receptors in the skin provide us with tactile information about qualities like the position, shape, texture, pressure, and movement of things we come in contact with. They are often classified as either rapidly adapting or slowly adapting. Rapidly adapting receptors are activated when a stimulus is first encountered, but fall silent if the stimulus remains present. They are important for detecting things like movement. Slowly adapting receptors keep responding to a continuously present stimulus and are important for detecting things like the size and shape of objects.
So just to jump in here really quickly, I want to make sure it's clear. The rapidly adapting receptors, essentially they are they're sending a quick message and then they shut down. So they rapidly adapt to whatever the new thing is. If there's a new change or a change in pressure, so if you poked really fast, right? So you're poking, and that is an initial sudden change, and then it moves. That rapidly adapting will send a signal as it is being poked and then taking off. But a rapidly adapting one, if you poke your skin and hold it there, as you're holding it there and there's no change, it's not going to be sending that signal. The slowly adapting one, it's going to be still sending that signal. So I'm still pushing on my hand right here. It's the slowly adapting receptors are still sending a signal. And then the rapidly adapting ones, as I remove that pressure, they will again send a signal. So putting those things together is one way that we can understand what is going on, what we're touching, what we're feeling. Is it rough? Is it smooth? Those kinds of things.
And are important for detecting things like the size and shape of objects. Touch receptors also vary in the size of their receptive field, or the area they can detect signals from. Having many smaller receptive fields typically allows for better tactile discrimination than fewer large receptive fields. There are four main types of touch receptors found in hairless skin, like that of the hand. Merkel's discs are slowly adapting receptors that have very small receptive fields and high spatial resolution. They are especially dense in the fingertips and are best suited for processing information about shape and texture. Meissner's corpuscles are rapidly adapting receptors. They have relatively small receptive fields, but their spatial resolution is inferior to that of Merkel's discs. They are especially effective at transmitting information about movement between the skin and another surface. This can be used to sense texture and to detect if an object is sliding past the skin, which is important for maintaining grip. Pacinian corpuscles are rapidly adapting receptors with very large receptive fields. They are thought to be most effective at transmitting information about vibrations objects cause when they are contacted or grasped by the hand. This information may be important for the use of tools. Ruffini's endings are slowly adapting receptors with large receptive fields. They are poorly understood but seem to respond most to skin stretching, like what would occur with the movement of the fingers. This information might be especially important in generating awareness of finger and hand position.
Right, so in that video, we get a little sense of where all of these are. Let's dive into them in a little more detail. We're mostly going to focus on these four over to the right. The free nerve endings, those are the ones that are more for pain reception, and we'll touch on them in more detail in a little bit later. So starting off, we will focus on how do these mechanical senses work? What is actually happening biologically for a mechanical sense? So the way that these mechanoreceptors work, the actual mechanism to get that mechanical sense, is from these PIEZO or piezo channels. It is a mechanical gate on cell membranes to send an action potential. So here we see a little diagram at the top, and we see another diagram below. This is the level of picture that we can get of these channels right now. So this is the molecule. This is from a view from inside the cell of these two different mechanical gates. And so when it's closed, it doesn't let any ions through. When it's open, it lets ions through. The thing that makes it open is some mechanical force on the actual cell membrane. So on the the receptive piece, you could think, the receptive piece, you could think of it as a specialized dendrite that has these receptors on it, these special gates. And so this is what they look like. They have this like propeller kind of shape, and they come together and let ions come down through the middle when they are open. But again, they open because there's something touching on that membrane. So it's literal physical interaction, physically pushes it open and lets the ions in. So here is a little video on that happening.
As I already hinted to you, this occurs in real proteins too. Let's start with mechanosensitive channels. So mechanosensitive channels, these are ion channels that David will likely talk a little bit more about later. They're usually surprisingly simple. These are typically just channels that are very large holes that open up to let water through in cells. And one of the reasons to let water through in cells is simply to adjust pressure. If there is too high...
Just to be clear, this is the same type of mechanical gate. Even though they're describing this in a different part of the body, the way the gate works is still the same way.
Would rupture. On the other hand, the channels, these holes can't be open all the time because then the cells would be leaky. So what we'd have are channels that under normal circumstances, they sit embedded in this bilayer up here and they are closed. They are relatively thick actually, so they're not super happy in the membrane and the membrane is not super happy with them. Do you see here how the membrane has had to become slightly thicker around the channels to be able to host them? What then happens is that if I start to stretch the membrane, why would I stretch the membrane? Well, if the pressure increases in the cell, eventually this will lead to pressure building up on the membrane, right? That's going to lead.
So again, in our purposes, the channel is opening to let ions in rather than other molecules like water. So in our case, these are specialized receptors at the end of the receptor to let in ions.
To a tension in the membrane. As this tension builds up, some of these channels will now start to open. Do you see the channel on the right there opening up? So the channel itself will undergo a conformational transition, but this is really caused by when this membrane is stretched out, the lipids will have to become even thinner. Again, the volume of the lipids is roughly constant, so if I want the area to be larger when I'm adding tension, that means the thickness has to go down. Well, then the protein will have to adapt to that. Otherwise, it would eventually no longer be in the membrane. The way these proteins adapt to it is that when they become thinner, the actual channel opens up. As we
So that gives you a little bit of a sense of this happening, and I've got another video to show you as well. So this is an animation based on the basically molecular scans that they did, basically taking a lot of various pictures of this structure from different like slides of cells that they had and kind of putting that together to see, well, what is actually happening here. So this is the outside of this neuron. This is the inside of this receptor neuron. So it's pretty cool to see those very small movements of these proteins, these channels to let in ions. So back to that, when there is actual physical movement, touching, scratching, cut, pressure, these will open, let in those ions, and start the action potential. All right, so that's what we see in the Merkel discs, the Meissner's corpuscles, the Ruffini endings, and the Pacinian corpuscles. All of them are using this receptive process. So there's the physical change makes them activate, but each of them activates slightly different. The structure of the receptor is a little bit different. It's more attuned to stretching forces or pressure, or it activates quickly or it activates slowly, right? So these differences lead to us putting those together in our brain to get a sense of what we are feeling. So the next one, the first one that we'll really focus on is the Merkel disc here. So it's really close to the surface of your skin. So it's got a finger right there in the picture. You can see its receptive field is pretty focused. It's really focused. It is slow to adapt. So this means that they will continuously activate to a force. So if I'm poking my fingertip, well, the Merkel discs are continuously active. They keep firing and firing and firing and firing because it still has that pressure there. So that means it's slow to adapt. It's slow to change to that pressure. Right? And so that's those Merkel discs. They're way up here near the surface of your skin. And so that can give you a really fine-tuned sense of touch. So if someone is reading Braille, for example, they're using these Merkel discs in that skin to be able to tell, well, what are these letters as they're reading the Braille. So Meissner's corpuscles are about that same depth, but these are a little bit different. They have a little bit larger receptive field. You can see kind of that image here. The receptive field is a little bit bigger, and also they are a fast to adapt cell. So if I'm pushing here on my fingertip, well, the Meissner's corpuscles, as I start to press, they'll activate, but then while I'm holding that pressure and not changing it at all, they're not sending a signal. So they're quickly adapting to change, and they're stopping their signal. So I push, it sends a signal, and then it stops sending a signal. Then if I take that pressure off my finger, it will say, hey, I've noticed a change again. And it'll send a quick signal and then stop sending that signal. So see those Meissner's corpuscles plus the Merkel's discs, they have pretty small receptive fields. They're close to the surface of the skin, but they're both doing it differently, right? So the Merkel's disc is sending a consistent signal like, I feel something, I feel something, I feel something. Versus the Meissner's corpuscle, it's like, oh, I felt something, and now I'm not signaling anymore. So putting those things together can help us get a better sense of what it is that we're touching or rubbing our finger over. If I've got my keyboard right here, I'm trying to find the little nubs to like where your pointer fingers are supposed to be in the proper keyboard position, right? So I am using those receptors together to figure those things out. All right, now we'll go into some receptors that are further more deep under the skin. So this is pretty deep under the skin surface. It's called Ruffini endings. They have even larger receptive fields. So it's getting data from a lot larger area on your skin. Also, these ones, just like the first one we talked about, they are slow to adapt. So they are continuously activating. So this Ruffini's ending, I might be getting, if I'm poking here versus a couple millimeters down on my finger, I might still be talking to the same Ruffini's ending or a couple of the same Ruffini's endings, right? And so like the video mentioned, these are also related to stretching and pulling. So if your knuckle, it's stretching, it's pulling, it can give you a sense of where your finger is in space, and that stretching and pulling can give you a sense also if this is something bigger or smaller, and being further under the skin, it's not getting as fine detail of like texture kinds of things in your touch reception. But again, these are slow to adapt. It's one that's just kind of continuously firing when it's having that pressure or stretch on it. Now the Pekinian corpuscle. This is even deeper. This is the deepest touch receptor that we have. It's basically either at or just under the level of dermis and subcutaneous fat. So it's like right there. You can find them a little above, a little below, but it's above like muscle and bone. Right, so we've got your skin, you've got a little bit of fat that's kind of like cushion all around your body, that kind of thing. It's at like that line essentially. So it's pretty deep there under the skin. It is a little bit finer of a receptive field than the previous one, the Ruffini ending. So it's a little smaller receptive field, but it's still a much bigger receptive field than, say, the Merkel's disc or Meissner corpuscles. So going back to these, so they are really deep. They're a pretty large receptive field, and they are also different in that they are fast to adapt. So I've got pressure here, the Pacinian corpuscles, Pacinian. I think I've only read it. I don't know that I've heard anybody else say this one. Receptor, uh, it's fast to adapt. So as you're pushing, it's not sending a signal anymore, right? So I'm still holding a consistent pressure on my finger. Well, these are not sending that signal while there's that consistent pressure, but when I release, they'll send a signal again saying, hey, I noticed a change. Right. So having these at different layers of your skin, having different receptive fields, having slightly different mechano reception to them, right? So whether it's stretching, whether it's just pressure, that kind of thing, that helps us put together all of these and figure out what exactly is happening. It's kind of similar. Hopefully, you're noticing a similarity between other receptors, what we've talked about. So, depending on the order of things in the class, you probably recently talked about vision. So, rods and cones are some of the main receptor cells, but in addition to those, we're using the horizontal cells and the bipolar cells and the ganglion cells to put information together in different ways with different receptive fields. Same kind of thing here. We are taking the information from these different touch mechanoreceptors in our skin to be able to figure out, well, what am I feeling? Did I just get a cut on my finger? Am I poking my finger? Is it a really sharp poke with like a needle? Is it a tattoo? Like trying to figure out what is going on. Or is this a word in Braille? Is this sandpaper? Is it something smooth? All of these are working together and giving the different information, and we learn over time like what those things are because we can put the information from these multiple receptors together. Kind of like we've got to practice using our eyes to figure out what things are. We're practicing using them early on in life as well. So this is a really good detailed video about these receptors that we'll go over. So it's a lot of repeat, but if you want more additional detail, this is a really good video for that.
The sense of touch, or tactile sense, is the perception of objects that come into contact with the skin. Basically, mechanical stimuli such as pressure, stretch, vibration, or movement cause physical deformation in tactile receptors, which are essentially nerve endings of sensory neurons. The deformation typically leads to opening of ion channels, allowing ions to flow in or out of the cells, resulting in cellular depolarization. If the stimulus is strong enough, action potentials are generated and sent to the brain. There are several types of tactile receptors located in different layers of the skin and deeper tissues. Anatomically, they can be encapsulated or unencapsulated, meaning they are wrapped or not wrapped in connective tissue, respectively. Functionally, they can be rapidly adapting or slowly adapting. Rapidly adapting receptors generate action potentials when first stimulated, then quickly adapt and reduce or stop generating signals even if the stimulus continues. They respond to changes and therefore detect movements and sequence of events rather than static objects. Slowly adapting receptors, on the other hand, keep generating signals for a longer time. They carry messages about steady pressure on the skin and sense objects' texture, edges, and shapes. Touch receptors also differ in their sensitivity. Some respond to light touch, others sense only heavy pressure. A sensory neuron receives signals from an area called its receptive field. Any touch within a receptive field stimulates one sensory neuron, transmitting one single signal to the brain. Being touched at 2 points within a receptive field would feel like a single touch. On the skin of the back, 2 points of contact centimeters apart may feel like one touch, while on a finger, 2 contact points just 2 millimeters apart can be felt separately. Receptors with small receptive fields are more sensitive. They respond to lower frequencies of vibration. Receptors with large receptive fields typically respond to higher frequencies of vibration. A touch sensory pathway involves 3 neurons. Touch receptors are first-order neurons. Their axons form sensory fibers that enter the spinal cord via the dorsal root of spinal nerve. Inside the cord, first-order neurons synapse with second-order neurons either near the point of entry or in the medulla of the brainstem. Second-order neurons then cross over to the other side of the cord before ascending to the thalamus. This is how sensory information from the left side of the body is transmitted to the right side of the brain and vice versa. Third-order neurons conduct the information from the thalamus to the sensory cortex. Sensory neurons from the face and head follow several cranial nerves, mainly the trigeminal nerve, to the brainstem, where they synapse with second-order neurons, which decussate and continue to the thalamus. The sensory cortex is spatially organized so that its adjacent areas represent neighboring regions of the body. The size of the cortical area representing a certain body region is proportional to the number of sensory receptors it has. Thus, sensitive regions with high density of receptors, such as fingers or face, have a larger cortical representation than the less sensitive trunk.
The one thing I'll bring up here at the end is, one, looks pretty funny, but also notice how much brain matter is relevant or used for your sensory area, for your hand and your fingers, as well as your face, your mouth, like putting food into your mouth, feeling it there. So this is one of the reasons that young babies like to put things in their mouth. So make sure that it's not like a Lego or something. Make sure you have like large toys that babies can play with, because they have a lot of sensory receptors, these same mechanoreceptors in their mouth and the skin there and the tongue, those kinds of things. So they get a lot of information about these things. So make sure for babies, you've got toys that they're not going to be able to choke on and swallow or like hurt themselves on, because there are more receptors. They learn very quickly that they can learn more about an object by putting it in their mouth. All right, so that was the review of all of these receptors. The next one that we will talk about is pain, and the pain receptors specifically. So pain receptors are an interesting thing. They are a little bit different than what we see with the other touch receptors in the body. So these are called free nerve endings, or nociceptors. And what it means by a free nerve ending is literally just that there are, you could think of it as dendrite branches here in the skin that are just around, that they're just like hanging out. They're kind of spread throughout the skin, just random dendrite branches that are available to detect any kind of mechanical pressure, thermal change, chemical change, and we also have some Usually it's one single receptor that it does, either the mechano, thermal, chemical, but we do have some that do all of these, that responds to all of the signals, so polymodal receptors. And so they're just kind of hanging out there throughout the skin, close to the surface level, but also kind of like throughout the surface. So they go a little bit deeper too. And they're there ready to get any kind of sensation. So they work a little bit differently than the other ones that we were just talking about. If there is an injury, they can respond directly to that injury. So if it actually cuts or damages the nerve itself, it can trigger an action potential in and of itself. But sometimes what's happening is what is described on this slide. So the injury happens and other tissues around that injury start to react. So in this case, it's a cut. So a mechanoreception thing happens. So you have damage, and now you have cells, part of your immune system and your blood cells. They start to act on that tissue damage. And that action on the tissue damage, trying to repair it, bring in materials, things like that, essentially that stimulates these nerve endings. So it lets them know, oh, hey, there are the substance P, CGRP. These are in the system as your body is basically trying to work on this wound, and that activates these receptors. So this can also lead to things like swelling, redness, the sensation of heat. You might have had that if you've had, say, like a cut or an itch or something that you're allergic to. Sometimes your body can produce like a little more heat because it's pushing more blood to that area. And so all of these processes at the same time can send a signal to these free nerve endings. Essentially, these dendrites that are kind of hanging out there, waiting for pain to come to give you a warning, can activate to these other body process of trying to interfere or trying to fix that wound. But again, it can also happen if you just directly cut those nerves, like damage them, that they can also send an action potential. So with this, they are also traveling to the spine. So they are sending out that message to the spine, but also they can tell other nearby axons too to do more of this process of releasing the SP and CGRP. So the substance P and the CGRP. So this is also how we do things like neurogenic inflammation in this area. So the neurons themselves, there's some inflammation around them, and it's part of the healing process. So that inflammation bringing more blood, bringing more nutrients and things like that. It can be a very helpful thing, but as we've seen with some of the disorders and problems that occur around the body, it can also lead to problems. But usually it's working well, and it helps us to heal from whatever kind of pain that we are experiencing. So another interesting receptor, in addition to that chemical and also physical, so the mechano and chemical senses, we also have temperature sensitive receptors on some of these nerves that are all throughout our skin. So interestingly, different gates are active at different temperatures. So we essentially evolved a bunch of different gates that are in our membranes, and if it's hot, you'll activate this TRPM3, and if it's colder, you'll be acting, or you will activate the TRPM8. And so the hotter you get, different receptors are opening up, and it can tell the body, one, what is the temperature of that water that you're trying to get your shower ready for, right? Is it too hot? Is it just right? What about the snow outside? Is it really cold? Is it really warm? What about the swimming pool water? What temperature is that? Essentially, a specific channel is open, or depending on like what the temperature, you might be kind of in between the sweet spot for a couple receptors. So maybe you have two opening up, and so that is how you know, like, oh, it's about this temperature, or this is the temperature I like my shower, that kind of thing. And they're literally opening up because of that temperature that it is, which is pretty cool. So you expose your skin to heat, and then you get the channel open for ions to come in. It sends an action potential through the spinal cord up to your brain. The brain receives that signal and tells you what to do. That being said, for something like an actual damaging heat, so if it's really hot, this message can go to the spinal cord and then just straight back to motor neurons to tell you to, say, pull your hand back from things. But if it's not that hot, you might get that signal to your brain and then decide what to do, right? If you're testing out, if your water's pretty close in the shower, you're like, oh, it's a little too cold or a little too warm. That's not like, hopefully for you, it's not at like a danger level for your skin. So it's not going to lead to your spinal cord sending the message back to pull away. All right, so that kind of message is going to go all the way up to your brain, where you can decide, nope, I do want it a little hotter, or I do want it a little bit colder. Versus if you touch the burning stove, well, that message is going to go to your spinal cord, back out to the motor neurons, and so you're already moving by the time it gets to your brain. All right. An interesting thing with pain is that we're using basically axons that are these sizes. So these two smaller sizes is what we see for a lot of the axons that are sending pain signals. And interestingly, the wider the axon, it leads to faster signaling. So some of our axons in our body can send signals up to about 300, even up to 400 miles an hour. But for pain, we're getting a lot shorter, a lot slower. So interestingly, though, with these fibers, the ones that are a little bit wider that are sending pain signals, we get the fast, like pricking pain signals through those versus like slow, dull pains are coming through the C fibers, which is kind of interesting that we're sending those messages through the axons up to the spinal cord, up to the brain, through these different sized neurons. And then the blue around here is the myelin sheath, essentially, is what it's showing there. But yeah, these fibers are sending the like sharp, fast pricking pains, and these C fibers are sending the dull, slow pains. So here is a good video on the path of these neurons, where they are sending the signal through the spinal cord. Your book does cover this, but I think this video is a good one just to kind of review which areas, how they are sending the message up to the brain. And then we'll watch a funny little one about a chemical pain sense of capsaicin.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss pain and the anterolateral system. Pain begins at nociceptors, cutaneous receptors that are specialized to detect noxious stimuli like extreme pressure, very hot or cold temperatures, or tissue damage. When activated, nociceptors send a signal to the spinal cord. The signal will be sent to the brain on pathways that make up what is known as the anterolateral system. The anterolateral system consists of 3 major pathways: the spinothalamic, spinal reticular, and spinomesencephalic tracts. The spinothalamic tract can be considered the main pathway for transmitting pain information to the cerebral cortex. It is involved in the awareness and recognition of where in the body a painful stimulus is occurring. Neurons in the spinothalamic tract carry pain information from nociceptors to the spinal cord, then immediately synapse with a second neuron that quickly decussates, or crosses over to the other side of the spinal cord before traveling up the spinal cord. These fibers proceed to the ventral posterior lateral nucleus of the thalamus, where they synapse with another neuron that carries the pain information to the somatosensory cortex. The function of the spinal reticular tract is not very clearly defined, but it may be involved in changes in the level of attention in response to pain. The fibers of the spinal reticular tract follow the same path as the spinothalamic tract, but they also project to areas in the reticular formation, which is a network of neurons found throughout the brainstem that are involved in a wide range of functions, including attention and consciousness. The spinomesencephalic tract, also known as the spinotectal tract, plays an important role in the control and inhibition of pain. Fibers in the spinomesencephalic tract follow the same path as the spinothalamic tract, but they synapse in an area of the midbrain called the periaqueductal gray. When neurons in the periaqueductal gray are stimulated, they can activate a natural pain suppression system that involves the release of endogenous opioids, which are substances that are produced by the body and have pain-killing effects. The release of endogenous opioids and other neurotransmitters leads to the inhibition of the pain signal in the spinal cord and naturally produced analgesia.
All right, so that was our little video on the paths that the pain takes into the brain. Now we'll watch a funny little one. We're not going to go into a ton of detail on the chemical sense of your skin, but it's kind of the same thing. You've got receptors that detect chemicals. So if you are allergic to something, or if there's a chemical cleaner that gets on your skin, those kinds of receptors will react to that and send that pain signal. One example of one of those types of receptors is one that's sensitive to capsaicin, so spiciness. And an interesting thing that, well, I think it's interesting, is that birds have far fewer of those receptors, so that they basically cannot taste spiciness. Something that's really spicy can still make their intestines unhappy, but they can't taste it. It's not a painful thing to eat the hottest pepper. So that's what this video is about.
It is often said that birds do not experience the spiciness of foods. But is this actually true? To verify if birds really cannot taste spiciness, we conducted an experiment at the Premium Parrot Cafe's Bird Lounge. Upon entering the Bird Lounge, the parrots greeted me warmly. With their assistance, we set up our experiment. We chose the Chunyang chili, known as the spiciest chili for this test. Before giving it to the birds, I sampled the chili myself.
What?
It was so spicy that it burned my tongue. The burning.
So I just want to point out that something spicy, burning your tongue, basically that's some of those reactions that we were talking about of your body's system send to send in other cells, to send additional blood flow, to help with any kind of pain that you're getting, will lead to that feeling of burning. I mean, the pain receptors are feeling the burning too, but it can actually get hot because of that that inflammatory response.
My tongue. The burning sensation we feel from chili is due to a component called capsaicin. Capsaicin triggers sensory receptors that detect pain and temperature, not taste, which is why it causes a sensation of pain and heat. It is believed, however, that birds hardly react to the spiciness of capsaicin. To see this firsthand, we offered a hot chili pepper to a parrot. First, I handed it to an impressive parrot named Luffy. He held it with his foot, bit off the stem, and ate it without any sign of discomfort. Another parrot managed to eat it as well, even getting several seeds stuck in its beak. The reason birds handle hot chilies so well is that their sensory receptors are different from those of mammals, making them largely insensitive to capsaicin. Therefore, they do not feel the heat from even pure capsaicin powder. This tolerance allows birds to consume even hotter chilies than we can, which is crucial for the chilies themselves. When you slice a chili open, it's filled with seeds, right? Plants produce fruits, primarily to disperse these seeds. Mammals typically chew their food thoroughly and break it down extensively in their digestive systems, often destroying the seeds in the process. Birds, lacking teeth, do not chew their food finely, and their quicker digestion often means seeds pass through their systems intact. Furthermore, birds' ability to fly over vast distances enhances their capacity to spread seeds widely. It appears that chili peppers have evolved to be eaten by birds rather than mammals by producing capsaicin, benefiting more from birds in spreading their seeds.
So just a funny little fact that I wanted to share. While humans have receptors for capsaicin, and we feel that as it's one of our pain receptors, so it's going to the same pain areas of our brain, birds, they have very few, if any, of those receptors, so they don't get spicy. They don't taste that as spiciness. So kind of a funny thing. So with this, let's cover some of the pain categories that exist, and also some of the methods that we use to relieve pain. Before we continue on, I actually want to mention most of the science videos that I share in my lecture recordings, it's usually not a problem. It's like it's free use. I can use the video, but that one that we just watched about the birds, it is possible that the video just wasn't there. If that ever happens, and it's usually because it was taken down by a copyright strike from YouTube, and I could go through the process of fighting it, being like, "This is fair use. I'm using it appropriately." But that can take a long time. So if a video's ever gone, it's just because it's been removed during that process. Just go to the link in the slides and you can watch the video if it's not actually playing throughout my lecture. All right, but yeah, that funny one of the birds, it's kind of funny. Birds evolved, or peppers evolved to be able to attack mammals that are going to chew up their seeds too much, versus birds who can eat the seeds and poop them out quickly without damaging the seeds and spread the seeds further. So these peppers evolved to hurt mammals and not birds, which I find pretty interesting. So, for pain, we have different categories of pains. Nociceptive and neuropathic. So nociceptive is we're activating those nociceptors directly by that damage to them or the inflammation around them. And then neuropathic is injury to nerves. Sometimes you get like a burning electric or tingling sensation because the nerves are sending a pain signal. So that is two of the main ways that we feel the pain, or that the pain signals are coming in. So here in the brain, these are some of the main areas that are relevant for feeling and experiencing those sensations of pain. They're in the temporal and frontal lobes, in the limbic system, as you've probably seen up to this point. This middle area of the brain has lots of signals coming through it for all different parts of our body, including the pain senses. But in addition to this, these are centers that can also send to emotion centers of the brain too. So we can have emotional responses to stimuli. You might have gotten hurt and felt sad, or hurt and felt mad, or something like that. That's part of this process. So that primary somatosensory cortex, the graphic is a little maybe unintuitive at first, but this is the gray matter area, that wrinkly portion of the brain where those sensations are going to. Here's the white matter on the inside. So yeah, that green is the gray matter. But yeah, the pain, itch, other senses, they're going through the thalamus and to that somatosensory cortex, but at the same time, they're going to the hypothalamus and the hippocampus. So for things like memory, the cingulate cortex, the anterior thalamus, you're getting a body response. So the both those things like inflammation, but also you might have racing heart or cooling down, and then also that emotion feeling at the same time. So all of these are processes that are happening throughout the body and the brain through this kind of complicated feedback loop that is very fast because these neurons can send signals very quickly. So for pain relief, one of the main ways that we try to relieve pain are with NSAIDs or NSAIDs. They help reduce inflammation, which can help reduce and that it reduces inflammation all over your body, unless you have like a direct like injection of it somewhere. If you're taking an ibuprofen, I think that's what these ones are here. If you're taking an ibuprofen, it's reducing inflammation all throughout your body, which includes the areas that you've had injured, and you're getting those pain signals being sent because of the damage and trying to work on the damage. So it is reducing that inflammation, so it's also reducing that pain. Other things that really help: physical therapy. So relaxing or strengthening muscles around an injury can really help. So physical therapy is one thing I definitely recommend for anybody who has any kind of pains that came from an injury, even chronic illnesses, People use physical therapy for things like even arthritis, so strengthening muscles to move correctly, move in a way that doesn't hurt and that you're not going to hurt yourself more. You might have experienced this in the past, that maybe you stubbed your toe or hurt your ankle, maybe twisted it like a sprain kind of thing. And then over the next few days, you realize your other leg is hurting because you're walking differently. You're favoring the injured leg. And so now you are injuring another part of your body through overuse and using it in the wrong angle, that kind of thing. That can happen pretty often, especially for someone who has had, say, a dramatic injury, a chronic illness, or chronic pain, um, something like an accident, arthritis. They will often try to adjust their walking on their own or their movement on their own in a way that leads to more pain. Another thing that we've seen some evidence for is acupuncture. It seems to work probably for the same gait control theory as some of these other pain relief methods that we'll talk about in a minute. So sending different signals can go on along those same axons that the pain does and send an inhibitory sense to the pain signal. That's essentially what's going on with the gait control theory. We've got a video and a little bit about it, but before we get there, those are some of the over-the-counter or some of the things that you might do first as a first line of defense. Another thing that's often prescribed is opioids. So we as humans, we produce our own endorphins, and endorphins are an opioid. We have opioid receptors because we do use opioids throughout our central nervous system. So there's three main receptors in the central nervous system that opioids can bind to. And these evolved over time for our natural endorphins, like natural body processes. But if we can flood them with more opioids, that is what relieves the pain. It decreases those sensations. It even tells your body to stop sending so many pain signals as well. So that's why they can be really effective, but they can be very addictive as well. So pain relief, in a normal kind of pain response, the presynaptic neuron is generating a large and wide action potential. So there's some kind of pain, there's a cut, you've been cut. So your presynaptic neuron is generating an action potential, and the postsynaptic neuron depolarizes from that. For morphine, essentially, it's reducing the presynaptic action potential. It suppresses that. It opens up additional potassium channels to the postsynaptic neuron to hyperpolarize it. Essentially, this means that the activation of that neuron that's trying to send pain is reduced. So it's not being as active. It's not sending those signals. And the neurons on the midway path are also inhibited from sending pain signals up as well. Cannabinoids also do pain relief. So it's same kind of thing for other neurotransmitters made from fats. It's pretty much what they're doing. Just if you're using a cannabinoid for pain relief, it's putting more of those into the system. So your neurotransmitter release and again, for neurotransmitters made from fats and cannabinoids specifically, they're coming from the postsynaptic cell. So most of the time we're talking about a neurotransmitter leaving from cell A to talk to cell B. In this case, we're actually having these neurotransmitters come from cell B and then activating a receptor in a cell A. And so with that again is leading to different communication between the cells, and it helps to make it so it's not as responsive to those pain signals. So let's watch that little video I was alluding to about gait control theory, because it's relevant to acupuncture, but as well as some of the other pain relief methods that we'll see in just a moment.
The gait control theory of pain states that the transmission of pain signals can be modulated at the spinal cord level by non-painful inputs as well as descending signals from the brain. Non-painful inputs typically close the gate to painful inputs, reducing pain. Pain sensation is mediated by pain receptors or nociceptors, which are essentially nerve endings of first-order neurons in the pain pathway. The axons of these neurons form small diameter nerve fibers of two types: fast fibers A delta, responsible for the initial sharp pain perceived at the time of injury, and slow C fibers, responsible for a dull, longer-lasting pain. First-order neurons travel by way of spinal nerves to the spinal cord, where they synapse with second-order neurons, which, together with third-order neurons, carry pain signals to the brain. However, according to the gate theory of pain, perception of pain is not simply due to stimulation of nociceptors. There is a so-called nerve gate located in the dorsal horn of the spinal cord that controls the passage of pain signals to the brain. This nerve gate consists essentially of interneurons that inhibit second-order neurons, thereby stopping or reducing signal transmission. When nociceptors are activated, they stimulate second-order neurons and, at the same time, inhibit the inhibitory interneurons, thus opening the gate, allowing transmission of pain signals. Now, if the area is also stimulated by other non-noxious stimuli such as touch, pressure, or changes of temperature, a different type of nerve fiber, large diameter A beta, is activated, and this reactivates the inhibitory neurons, stopping the transmission of pain signals. This mechanism underlies the pain-relieving effect of skin rubbing or heat or cold packs. It is also the basis of pain treatment procedures such as transcutaneous nerve stimulation, which delivers a small electrical current to activate non-nociceptive receptors in the skin. The brain can also send signals to the spinal cord to modulate the pain gate. Examples of such signals are endorphins, morphine-like substances released by the brain in response to pain. In addition to lowering pain perception by the brain, endorphins reduce transmission of pain signals between the first and second-order neurons by two mechanisms: preventing the release of a key neurotransmitter, substance P, and inhibiting action potentials in postsynaptic neurons. Production of endorphins may be induced by a number of factors, including pleasurable activities, excitement, meditation, laughter, and vigorous exercise. Endorphins' action underlies the pain-relieving effect of physical exercise, positive state of mind, and explains, for example, why athletes may not feel pain from an injury until the exciting game is over.
So hopefully with this example, you get a better sense of how these pain signals can be inhibited. So a little better understanding. I like that they made the myelin nice and blue for it having an ice pack on it here. But yeah, ice packs, rubbing. It makes me think of the Peter Griffin meme. He's bumped his knee and he's sitting there rubbing his knee, rocking back and forth. That can actually inhibit the pain signals. So if you go to a physical therapist for an injury or even not, if it's something like arthritis, oftentimes they will use like a combination of ice packs, maybe some heat or pressure or massage or something like that to try and help reduce those signals of pain, and the idea is this gate theory. So the gate control theory, they are inhibiting that signal here. And you can do that through your own endorphins, like exercise, things like that. So doing exercise, doing something that you enjoy can help your brain to produce and release more of those endorphins. So you're getting that natural morphine-like effect in addition to these pieces. So that's a lot of what physical therapists are going to be doing is one, helping you to move correctly so you don't re-injure or injure other parts of your body, and also relieving pain very directly by how your body is working on these things. So really good field. After a motorcycle accident, I went to physical therapists for it was a year and like 3 months. It was a long time, but they were great. They did a really good job helping me through that whole process. So I recommend it to everybody. We also got to see in these videos earlier on in the quarter, the person who had the really severe stroke and lost a portion of their brain because of the death of those cells, and that physical and occupational therapy helped them get back a lot of their functions that they had. But yeah, that is the gate control theory of pain. So other things that use this gate control theory, you can use transcutaneous electrical nerve stimulation. Basically, you put an electrode on the skin, or you can even put it under the skin in a medical kind of context. You wouldn't do that on your own. You can get these kinds of units to deliver a small electrical pulse that can help relieve the pain. Again, you're trying to put an interfering signal to inhibit the pain signal, right? So you're sending another signal that, just like rubbing or an ice pack, it's not a pain signal, it's something else that inhibits that pain signal. So that's what you're trying to do with the electrical nerve stimulation. You can also do this by directly stimulating the spinal cord itself. This one is going to be a much more involved process, but same kind of thing. It can give you tingling sensations or make you feel like there's tingling sensations, and that is working at that gate control to inhibit the pain signals from continuing. You might have it pulsing a specific amount of time at regular intervals, that kind of thing. Another thing that we've done is gone directly into the brain. So some of those videos mentioned the periaqueductal gray. We have done with electrical or magnets, some stimulation of those specific areas of the brain to change pain signals, to reduce them. So in this kind of case, again, this one, especially putting a probe into the brain, this is not done frequently. This is kind of a area of last resort, versus it would be very common to find someone at a physical therapist's office with this kind of TENS unit to give you that small electrical pulse on your skin. It would be very unusual to find someone who has the electric stimulation of a probe inside their brain, but it is a possibility. So another one that is more common because it's less invasive is transcranial magnetic stimulation. So this is used in a couple different disorders as well. But essentially, you have a big electromagnetic coil, generates a magnetic field, and it changes the electrical activity of neurons in a specific area of the brain. You can kind of target it to a pretty specific area of the brain. It's not like super targeted, but it's targeted enough. It's a little wider than a direct probe, but in this case, you can go in, basically go under the machine. It's kind of like a backwards MRI that's kind of hovering around your head, and you change the magnetic field in your brain, changes the neurons themselves, and that can help relieve some chronic pain as well. Some people might have to do this treatment a few times a week. Some might do it a few times a month. Some might just do it a few times and never have to do it again. We see that with a couple disorders, specifically mental disorders, treatment-resistant depression. This has been used for. Doing this a couple times kind of gets the neurons working in a different sequence and can let other treatments start to work. So it kind of just depends on the kind of pain that a person is experiencing, which one of these methods they'll do. Since this one is less invasive, it's a little more common, and it's more frequent now than doing a probe inside someone's brain. Because putting a probe in someone's brain, it's brain surgery, right? That's very invasive, versus a magnet. You come in, you walk out, no extra holes in your head from the time you come in to out for that kind of appointment. So this is a good review on pain in general. We've covered these topics in here, but if you want additional review, this is a good one in the slides. I'm not going to play it right now, but if you want an additional review, that is a good place to get it. The next thing we will do in this lecture is go through a little bit of Chapter 10. Hello students, this is Chapter 10. So this one we're going to do a lot briefer and not cover everything in the chapter, but there are a couple things that I definitely want us to cover. So the first is the motor system. How is the brain involved in motor functions? So we talked in Chapter 9 about sensations of pain and touch that are coming in to the somatosensory cortex. So now we'll talk about the section of the brain that's right next to it, and that is the motor cortex. So let's see how the brain is involved in this process.
The motor cortex is a region of cortex in the frontal lobe that is involved with voluntary movement. It is located anterior to a large sulcus called the central sulcus, and is often divided into two major regions, the primary motor cortex and the non-primary motor cortex. The primary motor cortex is found in the strip of cortex known as the precentral gyrus, which is located just anterior to the central sulcus. The primary motor cortex is arranged such that different parts of the region are associated with motor control of different parts of the body, and thus is said to contain a motor map of the body.
So just to jump in, just like we saw with the somatosensory cortex, there's specific sections of gray matter that are dedicated to specific parts of the body, the inputs of pain and touch coming from those specific parts of the body. We have in the motor cortex specific areas of gray matter that are dedicated to specific parts of the body. Again, we see a lot for face, mouth, throat. Facial expressions are a really important thing for humans. Eating and swallowing, also pretty important for humans. So, but it also doubles for things like kissing as well. So we've got a lot of area for our face. We also have a relatively large area for our hands to do really fine motor controls, play piano, clarinet, things like that. And then a lot less space for things like your legs.
Most of the neurons that travel from the primary motor cortex carrying signals regarding movement will enter one of two major motor pathways: the corticospinal or corticobulbar tracts. The corticospinal tract carries movement-related signals to the spinal cord to cause movement of the body, while the corticobulbar tract carries signals to the brainstem to cause movement of the head, neck, and face. The non-primary motor cortex is often divided into two main regions: the supplementary motor cortex and the premotor cortex. Although the functions of these areas are not well understood, it is thought the supplementary motor cortex may be involved with the execution of sequences of movement, the attainment of motor skills, and the selection of movements based on incoming sensory information. The premotor cortex contributes about 30% of the neurons that enter the corticospinal tract, but seems to be more active during the planning of, rather than the execution of, movements.
So another interesting thing with these areas, and we'll get to this in just a minute with the prefrontal cortex, is that they are also involved if you are imagining yourself doing something. So if you are envisioning yourself picking up a cup, if you are envisioning yourself driving a car, right? And like the motion, maybe you've just barely learned stick shift and you're imagining, okay, then my left foot is going to go to the clutch and my right foot is going to let off the gas. And then my right hand is going to go on to the shifter and move it such and such a way, right? If you're imagining yourself doing that, you actually do have some activity in the motor cortex as you are thinking and planning that ahead. It appears that for humans at least, and maybe some other mammals, it's a hard thing to try and like get clear evidence that a mammal is like planning that ahead or like envisioning that. For humans, it seems like we actually do increase our performance. We've had experiments where people do like 30 minutes of practice of, say, free throws or throwing darts are two specific studies I know of. And then we've also taken another group and had them do no practice at all. And so that's the control group. And then another treatment group that they envision themselves throwing a perfect free throw and throwing the dart the correct way after they know how to do it. Like it's not the first time ever doing it. They're just envisioning the proper form, what it'll feel like, that kind of thing. And we've seen that it's better than doing nothing, that actually envisioning it helps you pick up or improve those skills better than doing nothing at all. So the practice is obviously important too, but the envisioning plus practice seems to have a really positive effect at building some of the skills. And so some of these areas, the supplementary and premotor cortex, as well as the prefrontal cortex, are involved in that process. All right, so when we are thinking about something like throwing a dart or throwing a free throw, these decisions about what to do, where to move, those are coming from our prefrontal cortex. So these are a couple different images. This is basically information coming in to the prefrontal cortex that we then decide what to do with. This is information leaving the prefrontal cortex after we have decided what to do. Yeah, that's the first step, is I need to decide what to do, right? Am I going to go and grab my water bottle that's over here? Or am I going to go and grab my phone that's over here? I'm going to go and check my watch, right? So what are my decisions? That is where your motion first starts, is as long as it's not something that is like We talked in chapter 9 of the signal going just to your spinal cord if you're like burning your hand on a stovetop. That can then come to your somatosensory cortex, and then your motor cortex can say, okay, now do this. But in that kind of case, your spinal cord has already told the motor neurons. Most of our decisions about movement, our agentic movement, that's coming from the prefrontal cortex. So we have that prefrontal cortex, and we send that then. So we get this information from brainstem, from areas of our body, from our vision, from the sensory receptors on our skin, all over our ears to see what our balance is. And that is all going to our prefrontal cortex. And then we go, I'm going to grab my water bottle, right? Something like that. So when we decide that, that goes to the premortal cortex. So the dorsal, ventral, cingulate, supplementary, and they all coordinate, hey, what muscles should be activated? In the primary motor cortex, what should we be activating? So that's the first step. Prefrontal cortex says, I want to grab my water bottle. Premortal, premotor cortex then coordinates, hey, these muscles should get activated to do this job of grabbing the water bottle. And that information then goes through the posterior sensory cortices, the basal ganglia, and through the thalamic way station on the way to the cerebellum. So at the same time, you're sending signals to the basal ganglia, and you're sending signals to the cerebellum. Then, after you've sent them to the basal ganglia and cerebellum, the primary motor cortex also gets these information and sends detailed movement instructions down to the spinal cord. So this is all happening like in this kind of sequence. And that primary motor cortex says, okay, so I need to move my right hand over here, grab the water bottle, and bring it over and use it to drink my water, right? So that is coming from prefrontal cortex, going through the premortal cortices to decide like which muscles need to be activated. Does the shoulder need to activate? Does the left shoulder need to activate, right? If I was like leaning, maybe it would. Maybe I need to like balance myself, right? So the premotor cortex helps coordinate like these are the muscles that should be activated, send those signals through the sensory cortices to the basal ganglia and to the cerebellum, and also, hey, primary motor cortex, send those signals to move those parts of the body. There is also feedback loops from the cerebellum and from the sensations of like where your body is in space. So that proprioception, knowing like where your arm is in space. Is it already really close to the water bottle? Is it further away from the water bottle? That kind of thing. You're also getting that feedback loop as you are starting to do that movement. So these are the afferents to the prefrontal cortex. These are the signals coming into the brain. You have dorsal areas letting you know what the limbs are doing. And if you see dorsal versus ventral, think of like a dolphin. So like the dorsal fin is on the back, right? So like the top back part of the brain. That's the dorsal part of it. Ventral is the lower part, like closer to your jaw. So like if you were a dolphin, your face would be going that way. That's ventral side. And then the lateral side as well. But yeah, the basal ganglia is also sending information up to the prefrontal cortex saying, hey, this is what the body is doing. It's breathing, it's breathing hard or not hard, that kind of thing. So all of that's coming to your prefrontal cortex, and then you got to decide, hey, I want to grab my water bottle. Right? Maybe one of these signals is saying, hey, this person's mouth feels dry or their throat is itchy, right? So then I decide, oh, I want my water bottle. So when I do that, the prefrontal cortex helps to modulate the activity. It sends it to the basal ganglia, which also is going to the cerebellum. It's also going to the primary motor cortex. And there is a feedback loop between some of these to let you know where your body is in space. So this is very helpful, a very complicated system of multiple steps and feedback loops to figure out how to do something that's seemingly as simple as grabbing the water bottle to drink a sip of water. All right, so those are some of those main steps in that process. But again, it's all starting from the prefrontal cortex, deciding to do something and taking these steps through the process. Here we see again the premortal premotor area and the primary motor cortex, with the gray matter dedicated to specific parts of the body. Again, hands, face, swallowing, tongue, those kinds of things. Lots of gray matter, lots less gray matter for the trunk and the legs and the feet. So for someone who's doing, say, a trick or a gymnastics move, or trying to do something like pole vaulting, well, you're trying to plan, right? You are thinking about, this is what I've got to do. Your premotor cortex is deciding these muscles are probably going to be needed. Supplementary motor areas are planning, like, well, first you got to move the leg this way, then this way, then this way, right? If you're doing a backflip, first you've got to jump and throw your arms up, and then you've got to you don't have to throw your head back, but you jump, throw your arms up, and tuck your knees really hard, and that'll help you rotate. And then you've got to spot the ground, or you really should spot the ground, and pull your legs back out to land. Right? And then the primary motor cortex actually is going to send those signals to have those muscles flex and relax when they need to in that process of something pretty complex. So the basal ganglia is a really important part of this process. Again, the prefrontal cortex is sending messages to the basal ganglia. It's also going through the basal ganglia to the cerebellum. But let's look at what the basal ganglia specifically, its job and the feedback loops there is doing to then talk to the motor cortex and other parts of the brain.
The basal ganglia are a group of structures that are generally considered to include the caudae and putamen, which are collectively known as the striatum, the globus pallidus, subthalamic nucleus, and substantia nigra. The globus pallidus is further divided into internal and external segments, and the substantia nigra is divided into the substantia nigra pars compacta and substantia nigra pars reticulata. The direct pathway is a circuit in the basal ganglia, best known for its hypothesized role in movement. The direct pathway model involves glutamate neurons that project from the thalamus to motor regions of the cerebral cortex. These excitatory projections are thought to be involved with stimulating movement. Neurons from the globus pallidus internal and substantia nigra pars reticulata, however, project to the thalamus and maintain a steady release of the neurotransmitter GABA, which acts to inhibit the thalamic neurons and suppress movement. This mechanism is thought to be important in keeping unwanted movements from occurring. When we want
So before we continue, I just want to emphasize there that, like we've seen in some other sensory receptors earlier on, like in the vision system, some of them are sending like consistent inhibitory signals. That's what's happening in the basal ganglia. We're getting inhibitory signals to the thalamus saying, don't move, don't move, don't move, basically. And then we have to do other things in our brain to overcome that and get a movement to actually happen.
to make a movement, however, information about the movement is sent from the cortex to the striatum via the corticostriatal pathway. Glutamate neurons in this pathway excite neurons in the striatum, and the activated striatal neurons release GABA in the globus pallidus internal and substantia nigra pars reticulata, inhibiting the activity of these regions and stopping the inhibition of neurons in the thalamus that are involved with movement. This effectively opens a gate for movement to occur. Activity along this pathway tends to occur just prior to a movement, and thus has been linked to the facilitation of movement. The substantia nigra pars compacta is thought to modulate the activity of the direct pathway. Neurons from the substantia nigra pars compacta travel to the striatum via the nigrostriatal pathway and release dopamine in the striatum. One effect of this seems to be the facilitation of activity in the direct pathway.
All right, I'm trying to put these both on screen at the same time, so you've got that diagram from that video we just watched, and here's an actual cross-section of the brain where you can see these parts of the brain that they were just talking about. So this is the basal ganglia, we can see the striatum, the direct and indirect paths that are using these different parts of the brain, sending information back and forth in this loop to inhibit that, to inhibit movement and make it so that you're moving when you want to be moving and not moving when you don't want to be moving. So those are those little areas that are doing this process for helping us move and not move and get the complicated movement of a backflip or picking up a water bottle to happen. All right, so one thing I want to emphasize with this is that disinhibition. So the multiple sensory regions are converging from the cortex. So the prefrontal cortex, we decide to do something, and that's exciting. The caudate and putamen, and those are inhibiting the globus pallidus right here, and that makes it so it's disinhibiting the messaging to the thalamus, which means that we can then do a movement. So with this area of both the basal ganglia and the cerebellum are very important for motor sequences and muscle ensembles. So essentially, when we say muscle memory, what people are usually talking about there is like when I go to pick up my phone, I know where the buttons are, right? Like I know where the fingerprint sensor is. In my car that I've been driving for years now, I know exactly where like the volume knob is, right? So I have muscle memory. Well, really, that muscle memory is actual memories in the basal ganglia. So it's literally a pattern of activity, of activation of a specific set of neurons that go here in the basal ganglia, also the cerebellum, also the motor cortex, to then move to turn the knob in my car. So a motor sequence is like a timed muscle activation of playing a scale on the piano or playing a song on the piano or playing a section of a song on a piano. That's a motor sequence. You do this finger, this finger, this finger, this finger in this kind of pattern. Versus a muscle ensemble is a little bit different. It's like if I'm going to do a pole vault, well, actually, I've not done it for many years. I did that in high school. But you run down with your pole, you put it in the thing, you are running fast enough, and you drive your knee, and your pole bends, and that pole pulls you into basically changes your forward momentum to upward momentum, and then basically do a upside down push up off of the pole to jump over the little bar. So complex set of movements, but it's an ensemble, right? It's activating simultaneously. I've got to drive at the same time as I'm jumping. So there's a couple sequences there of ensemble muscles, like putting them together, right? If I am trying to do a something more simple, let's say I'm trying to lift a weight bar and do a curl, and I have one bar in both my hands, right? There's multiple muscles involved there, right? I've got to be having my core engaged. I've got to have at least both my biceps engaged. If I only pull one, well, that's going to be lopsided. It's not going to do what I'm wanting to do, right? I have to activate these muscles simultaneously to get a specific movement to happen. So something like kicking a soccer ball, you're taking a bunch of muscles at the same time. So you can put these together, right? A motor sequence plus a bunch of muscles, but we can also look at them separately as well. But yeah, that is involving this basal ganglia and literally specifically memories, a network of neurons that know how to play that scale or what muscles have to be active to be able to flip someone onto their back, right? Something like that. And the cerebellum is another piece of this puzzle. So it has tons of gray matter. It looks like a little brain. We've got the motor cortex way up here. We've got the basal ganglia somewhere around here. And essentially, what's happening here is the cerebellum helps coordinate the timing of things. So, how smooth the movement is, how fast or accurate it is, where is your body, right? If I'm going to be typing a word on my keyboard, well, my cerebellum is getting a message of, oh, my fingers aren't actually on the keys. They're like in the corner of the keys rather than the center of the keys. My pointer finger is not on like the home line of keys, right? Or if you are riding a bike, if you're tipped one way or another way, well, you're getting signals from your inner ear and also going to your cerebellum telling you where you are, where your balance is. You one knee up and one knee down, right? Like where is your body? And so this helps with that coordination. So not only do we have the prefrontal cortex, we have the prefrontal cortex, we have the thalamus, we have the basal ganglia, we have the cerebellum, we have the primary and they just left the supplementary motor area, the premotor area, and the primary motor area. All of these areas involved in our basic movements that we're doing day by day. So that is a big part of the motor system. The last thing I want to cover is how the muscles themselves work. How are the muscles actually activating from a motor neuron? So we're going through that whole process. We've got the prefrontal cortex, basal ganglia, cerebellum, motor cortexes. Those are all talking with each other and sending the signal to say, "Hey, biceps, move." At the same time, well, what actually happens when that signal gets down those neurons to the muscle cell itself? So let's watch this last video about how we get that muscle to move.
A skeletal muscle contracts only when stimulated by nerve impulses from a motor neuron. Without innervation, muscles become paralyzed and eventually atrophied. The axon of a motor neuron usually gives out many branches, supplying multiple muscle fibers. These fibers contract in unison when activated and constitute a motor unit. A motor unit can contain anywhere from just a few muscle fibers to thousands of them. Small motor units are found in muscles that require finer control. For example, muscles that are responsible for subtle movements of the eyes. Large motor units are found in larger muscles that require strength, such as muscles of the arms and legs. Muscle fibers of the same motor unit are usually not clustered together, but scattered throughout the muscle. This way, stimulation of a motor unit has a slight but uniform effect on the whole muscle, and not causing a small, localized twitch. The strength of a muscle contraction is determined by the number of motor units that are activated at one time. Even at rest, most muscles are in a partial contraction state called muscle tonus, which is maintained by alternating activation of a small number of motor units. The connection between a motor neuron and a muscle fiber is called a neuromuscular junction, which is basically a chemical synapse between the nerve terminal and a specialized area of muscle cell membrane called the motor end plate. When an action potential reaches the nerve terminal, it causes the release of the neurotransmitter acetylcholine into the synaptic space. Acetylcholine then binds to nicotinic receptors on the end plate. Nicotinic receptors are ligand-gated ion channels. Upon binding to acetylcholine, they open to allow sodium to enter the cells, depolarizing the cell membrane, producing the so-called end plate potential. An action potential is generated in the muscle cell only when the end plate potential reaches the threshold required to activate voltage-gated sodium channels located outside the end plate, in the neighboring membrane. When activated, these channels allow faster influx of sodium, further depolarizing and eventually reversing the polarity of the cell membrane. At this point, voltage-gated potassium channels open for potassium to move out, quickly returning membrane voltage to its original resting value. Once generated, the action potential spreads like a wave, thanks to similar voltage-gated ion channels located throughout the muscle fiber. The action potential also runs deep into the fiber via T-tubules to reach the sarcoplasmic reticulum. Here it activates voltage-gated calcium channels, releasing calcium from the sarcoplasmic reticulum into the cytosol of muscle cells. Calcium then sets off muscle contraction by the sliding filament mechanism. This mechanism is described in another video. Another important component of the neuromuscular junction is the enzyme acetylcholinesterase. This enzyme removes all acetylcholine molecules that do not immediately bind with the receptor, and those that are done activating a receptor. The enzyme action essentially terminates synaptic activation, giving the muscle time to relax and thus preventing continuous contraction that would result in muscle spasms. Substances that cause muscle weakness or paralysis do so by interfering with the function of neuromuscular junction. Botulinum toxin prevents acetylcholine release from the presynaptic side of the junction. Some other toxins attach to nicotinic receptors, blocking acetylcholine from binding but do not open the ion channel. Certain drugs lodge into the channel of nicotinic receptor, blocking the passage of sodium. All these substances prevent activation of muscle cells and cause flaccid paralysis. On the other hand, some pesticides inhibit acetylcholinesterase, preventing degradation of acetylcholine causing continuous activation of muscles. That's how they induce muscle spasms and cause spastic paralysis.
All right, so that is the last step. Well, one of the last steps. I definitely recommend kind of skimming the book of the chapter 10 of what happens from this point on. But yeah, once we have that information, we've decided to do something from our prefrontal cortex. We've gone through the basal ganglia, through the cerebellum, through the motor cortex, sent that signal down the neurons, all the way to the muscle cell itself to have that muscle cell literally pull on itself to contract the muscle, and doing that across a large muscle like a bicep makes it so your arm moves from this position to this position. Right? So that is the big multi-step process. And again, it couldn't happen this fast without neurons, that some of those interneurons can send messages up to like 400, basically like 300 miles per hour kind of thing. And so it's a pretty impressive system. We can get the sensory input of, say, something like pain or touch or something like that. So if I am feeling around in the dark for my water bottle or something like that, and I feel the cold metal on my hand or something like that, then I can decide in my prefrontal cortex, hey, let's pick that up and drink some water. So lots and lots of systems that are involved here. Again, we're not going in as much detail for chapter 10 as we did for chapter 9, but hopefully from the brain areas and also how the neuron is talking to the muscle cell itself, gives you a better sense of how your body is working and doing things. And hopefully now you also understand that muscle memory is literally just memory. So it's literally a memory in your brain, a network of neurons that knows to fire together, just like some of the memories that are strongest for you. That's a network of neurons that runs as the memory itself. So muscle memory is just memory. And it's not actually in your muscles. Your muscles are just these little things with a bunch of different proteins and tubules and all sorts of things together that do the job of contracting and not contracting. So it's really all down to your brain. So hopefully this is helpful for you to see that whole system of 9 and 10 together. And again, focus your reading on chapter 9 and skim over chapter 10. Definitely review some of the things in the activity for the week as well. It dives into a little more depth in that process of going from the prefrontal cortex to the basal ganglia, to the cerebellum, to the motor cortex, and then sending that message to the muscles. Thanks.